This is Yashveen. In this video, I'll be solving paper 4 variant 2 February-March 2022 IGCSE Biology. Question 1a, part 1. State the name of the gas exchange surface in humans. That will be alveoli. 2. State two features of the gas exchange surface in humans. So the gas exchange surface or the alveoli, they have a large surface area and they have a short distance of diffusion for gases like carbon dioxide and oxygen, since the alveoli are only 1 cell thick, and they have a good blood supply due to the blood capillaries, and as well as a good ventilation, which is the movement of air into and out of the lungs. So you can write any two of them. B. Figure 1.1 is a diagram of the gas exchange system in humans. So here you've got the labelings of trachea and the diaphragm. Draw a label line and the letter X on figure 1.1 to identify an external intercostal muscles. So here you've got the intercostal muscles. So here on the outer surface, I'll draw the label. To state the name of the tissue that forms C-shaped structures in the wall of trachea and state its function. So those would be the cartilage over here, and this keeps the trachea open and prevents it from collapsing. Three, describe the effects on the thorax of contraction of the diaphragm. Alright, so when this diaphragm contracts, it is going to move downwards, and as a result of that, the thorax or the part above the diaphragm will be allowed to expand in volume, and so as the volume will increase, the pressure of air inside the lungs or the thorax will drop, and after that the air will be drawn in through the nose. See table 1.1 compares the composition of inspired and expired air. 1. Complete table 1.1 by writing the names of gases B, C, and D. Alright, so gas B, we're supposed to name it, and we're given that its percentage is 21 in the inspired air and 16 in the expired air, and that is oxygen. Similarly, the gas, which is 0.04% of the inspired air, is carbon dioxide. And it's water vapor, which is variable, is of variable percentage in the inspired air, and this depends on the humidity of the environment, and it saturates the expired air. 2. For gas B and C, explain the differences in the percentages shown in table 1.1 between inspired and expired air. Alright, so gas B or oxygen, when inhaled, it will diffuse from the alveoli into the blood capillaries that lie next to the alveoli to be used for aerobic respiration. And so as a result of that, when it's used up, the concentration will be decreased in the expired air. As for carbon dioxide, it's produced as a waste product of respiration, and so it will diffuse from the capillaries surrounding the alveoli into the alveoli, which will then be exhaled, and so the percentage of carbon dioxide is going to be greater in expired air compared to inspired air. Here the key word is diffusion of gases. For example, oxygen will diffuse from the alveoli into capillaries, and carbon dioxide will diffuse from capillaries into the alveoli. So make sure you do mention that word. Question 2a, figure 2.1 is a photomicrograph showing the fertilization of one human egg cell. Describe and explain the adaptations of the cells shown in figure 2.1 that enable fertilization and the early development of the embryo to occur. So you're supposed to describe and explain the adaptations of sperm cells and egg cells. Starting with the sperm cells, they have an acrosome, which is a vesicle that contains enzymes that will dissolve the jelly coat which surrounds the egg cell, and so this will allow the genetic material present in the nucleus of the sperm cells to meet or fuse with the genetic material or the nucleus of the egg cells. which will eventually form the zygote. The cells also have many mitochondria in the middle piece of the cell, and this will provide energy to for swimming movements of the cell to transport it towards the egg cell. Sperm cells also have a flagellum, which aids in their swimming movement. Moving on to egg cells, they have a jelly coat which will harden once a single sperm has fertilized the egg, and this will prevent any more sperm cells entering the egg cell. The cytoplasm of the egg cell has yolk, which contains energy store. The gametes, both of the gametes, the eggs and the sperms, are haploid, or they have 23 chromosomes, so that when the zygote forms, it will be diploid, it will have 46 chromosomes. B. People can use artificial insemination or in vitro fertilization to increase their chance of becoming pregnant. 1. Outline the process of artificial insemination. So, in this process, a sample of semen is collected from the father or the male donor, and it is injected into the uterus. This is done close to the time of ovulation, which means that the egg is more likely to be present in the oviduct, and so this will increase the chances of fertilization. To outline how the process of in vitro fertilization differs from artificial insemination. So, in vitro fertilization, the gametes are collected from both of the parents, whereas in artificial insemination, the sperm sample is collected from either the father or the donor only. And in vitro fertilization, fertilization occurs outside of the female body, typically in a petri dish, and this will result in the production of zygote, which will eventually develop into an embryo, which is then implanted into the uterus of the female. 3. Describe the social implications of fertility treatments. In this part, you'll have to describe the pros and cons of fertility treatments. The advantages would include the fact that screening the embryo before introducing them into the uterus can reduce genetic disorders. So, prior to inserting the embryo into the uterus, you'd be able to see if the cells of the embryo have any disorders, and if they do, they can be discarded, and if they do not, then they can be implanted into the uterus. The benefits also include the fact that that fertility treatments allow infertile people to have offsprings. As for the disadvantages, the treatment can be very expensive and it can result in multiple births as well, and there's also the concern of ethical or religious issues with these treatments. Question 3a. A scientist investigated the effect of temperature on the mass of leaves picked from a tea plant, Camellia sinensis. 3 samples of leaves were picked and the mass of each sample of leaves was recorded. Each sample of leaves was kept at different temperatures for 4 hours. After 4 hours, the mass of each sample of leaves was measured and recorded again. The scientists then concluded the final mass as a percentage of the initial mass for each sample. Figure 3.1 shows the results, and in part 1 you're supposed to explain the results in this figure. So as you can see, as the temperature increases from 0 to 40 degrees Celsius, the final mass as a percentage of the initial mass is going to decrease, which also means that as the temperature increases, the mass of the leaves is going to decrease, and that's essentially because of the fact that as temperature increases, water will be lost from the leaves at a higher rate, and due to this water loss, the mass of the leaves will decrease. And in this answer, you're supposed to explain this with the details of the process of transpiration. As the mass of leaves will decrease, evaporation of water from cell walls of mesophyll cells will increase, and so this is going to be collected in the air spaces in the leaf, which will then be followed by an increase in diffusion of water vapor through the stomata, and this is an increased rate of respiration. 2. State one factor other than temperature that would affect the loss of mass from the leaves of a plant. The loss of mass, or in other words, the loss of water from leaves of mass. Here you can mention any factor which affects the rate of transpiration. I'll be writing humidity. B. Figure 3.1 is a photomicrograph of the tissue that transports water and mineral ions in a plant. So this is xylem. 1. State the name of the tissue shown in this figure. 2. Describe how the tissue in figure 3.2 is adapted for its functions in the plant. Alright, so it's got multiple factors, starting with how the hollow lumen of the xylem vessel will reduce the resistance to flow. The end walls of the cells that make up the xylem vessel have disappeared, which will also reduce the resistance to the flow. And the large cross-sectional area in xylem will maximize the volume of water that can be transported at any given time. You can mention any 3 points for the 3 marks of this question. C. Explain how mineral ions enter a plant. So remember that mineral ions are obviously dissolved in solution, and so they'll be absorbed from the soil by the roots. The roots are going to absorb these mineral ions by active transport using energy, as the mineral ions will move against their concentration gradient. So they are present in a large quantity in the soil and in a smaller quantity in the roots. Question 4a, figure 4.1 is a diagram of Vibrio cholera, the bacterium that causes cholera. 1. Describe 2 similarities and 2 differences between a palisade mesophyll cell and the bacterial cell shown in figure 4.1. So you're supposed to describe these similarities and the differences between a plant cell and a bacterial cell. So both of them who have cytoplasm, cell wall, cell membrane, and ribosome, and whereas the plant cell will have mitochondria and chloroplast, which the bacterial cell will not, and the bacterial cell will have a flagellum and capsule, which the plant cell does not. You can write any 2 of similarities and differences. To explain how the cholera bacterium causes diarrhea. Alright, so the bacteria will release a toxin which causes the secretion of chloride ions into the lumen of small intestine. And as the water potential of the lumen will decrease, water will move by osmosis out of the body, out of the cells in the body and into the intestine. And this water will be lost from the body in phases. As excess water is lost from the body and the cells are deprived of water, it will lead to diarrhea. B. A scientist tested the resistance of one strain of bacteria to different antibiotics. The scientist tested solutions of 5 different antibiotics, A to E. She soaked a paper disc in each antibiotic solution. So you've got 5 different antibiotic solutions where, and using them, you'll be soaking 5 different discs. The paper discs with antibiotics are placed in a petri dish containing bacteria on agar gel. On agar jelly, figure 4.2 is a diagram of the appearance of the petri dish after 48 hours. The shaded areas show where the bacteria grew, and the clear areas show where the bacteria did not grow. So as the antibiotics will kill the bacteria, they will prevent the growth of bacteria as well and cause the clear area, whereas in the shaded area, you've got the growth of the bacteria where they are not going to be killed. As you can see in the diagram, the clear area is the greatest around the disc, which is soaked in antibiotic E. So antibiotic E is going to be the most effective against this particular strain of bacteria, as the disc E is surrounded by the greatest clear area, which indicates that it has killed the most bacteria and the bacteria are least resistant to antibiotic E. On the other hand, the bacteria are most resistant to antibiotic A, as the clear area is the smallest around disc A, and this indicates the fact that the bacteria continue to grow and they are not killed by antibiotic A. Once the strain of bacteria used in this investigation causes a disease, Using the information in figure 4.2, explain why antibiotic E would be the most effective at treating this disease. So again, treating this disease would mean to eliminate the bacteria, and antibiotic E would be the most effective since it has killed the most bacteria out of all of the antibiotics A to E. The results in figure 4.2 show that this strain of bacteria is resistant to antibiotic A. 5 years ago, a similar investigation found out that the clear area for antibiotic A was the same size as antibiotic B, meaning that 5 years ago, the bacteria were not resistant to antibiotic A, and antibiotic A was just as effective in killing them as antibiotic B is now. And you're supposed to explain how bacteria become resistant to antibiotics. Alright, this happens when bacteria are continuously exposed to antibiotics. The antibiotics will act as selection pressure, and this will force them to evolve resistance. Continuous use of antibiotics throughout the 5 years has acted as selection pressure, and this will lead to mutation in the bacteria, which is essentially a change in the base sequence of the bacteria, and this will produce bacteria with an allele of DNA that causes resistance. So this new allele with a different base sequence of DNA will result in resistance to antibiotic A or any antibiotic that has been used. Now the bacteria with resistance will be able to survive as they won't be killed by this same antibiotic, and they will multiply to pass the allele for resistance to the offsprings. And over time, the frequency of resistant allele will increase in the bacteria, which will produce antibiotic resistant strains of bacteria. Describe how to minimize the risk of antibiotic B developing the same results as antibiotic A. One way to do this is to complete the course of treatment, which will ensure that all the bacteria are killed and they do not survive to evolve resistance to the antibiotic. The Arabian oryx and the northern white rhinoceros are both mammals. Figure 5.1 is a photomicrograph of an Arabian oryx, and 5.2 is the photomicrograph of northern white rhinoceros. Describe the two pieces of evidence visible in figure 5.1 and 2 that show that these animals are mammals. So both of them have external ears and hair or fur on their skin as the evidence. Different conservation methods are used to try to prevent species from becoming extinct. A population of Arabian oryx and a population of northern white rhinoceros were monitored. Figure 5.3 shows how the population size of each of these species has changed over time. So the cross points represent the number of Arabian oryx, and you've got their scale up on the left side, and the scale of rhinoceros is on the right side. They're represented by crosses that are circled. 1. Calculate the percentage increase in the number of Arabian oryx between 1990 and 2000, giving your answer to 3 significant figures, and you're given the space for working. You can see that in 2000 the number is 260, whereas in 1990 the number is 75, and you can calculate the percentage increase by subtracting 75, the initial, from the final number and dividing that by the initial number, multiplying the fraction by 100, and this will give the final answer as 246.66, which will round to 3 significant figures as 247. 2. Describe the data for the northern white rhinoceros in figure 5.3. Now, the word describe means that you are supposed to talk about the way that it changes or the graph is changing and not explain the reasons behind it. So again, the number of rhinoceros is represented by the crosses that are circled, and their axis is on the right hand side. You can see that the number of rhinoceros is going to increase from, this is 15 in 1980, and it's going to peak in around 2000 at the figure of 32. And then the number of rhinoceros is going to decrease from 32 in 2003 to the final number of 2 in 2020. He suggests the conservation methods that were used to increase the number of Arabian oryx between 1970 and 2000. This could include providing them habitats with protection, where hunting will be prohibited, and by using captive breeding programs which are carried out in zoos to increase the population size of endangered animals. They explain the risks to the northern white rhinoceros species as a result of its population size. So as the population size is very small, it might face inbreeding and reduce genetic diversity as closely related individuals mate repeatedly, and the rhinoceros will be less likely to survive and adapt to changes in environmental conditions, and as a result of this, their numbers will decrease further and they might become extinct. 6a, complete table 6.1 to show the names, functions, and sites of the action of 3 different digestive enzymes. Pepsin will catalyze the conversion of proteins into amino acids in the stomach, whereas trypsin will catalyze this conversion of proteins to amino acids in the small intestine, and maltose will break down maltose to glucose in the small intestine. Three enzymes are proteins. Figure 6.1 shows stages involved in protein synthesis. So in here you've got the DNA and a section of it which is going to code for protein, and by definition we know that a section of DNA which codes for protein is known as a gene, which is labeled as A. 1. State the name of the parts represented by the letters A and C. So as I mentioned earlier, A here is a gene, and C, as you can see, is the site of translation, so it is a ribosome. 2. Describe the events that occur during stage 2 in figure 6.1. So in stage 2, here you've got an mRNA, as it is a single strand compared to the double-stranded DNA, and it is leaving the nucleus through the nuclear pores. 3. State what determines the order in which the parts labeled B are assembled. Here we've got step 3 where B, or amino acids, are being assembled. So the question is asking you what determines the sequence of amino acids in a polypeptide or in a protein. This sequence is determined by the codon on mRNA, which is a triplet or 3 bases on the mRNA, since each codon codes for a single amino acid. Hence, the sequence of bases on the mRNA or the codon will code for the sequence of amino acids in a polypeptide or protein. See, the shape of protein is very important for its function. Explain the importance of shape for the function of an enzyme. Proteins, like enzymes, function with specific substrates that are complementary to the shape of the active site. This will allow the formation of enzyme substrate complex as the substrate binds to the active site of the enzyme, which will then allow the enzyme to function. We are done with this paper. Thank you for watching.